Hi everyone, so today in this video I'll be talking about the PCR and the primer design, where I will highlight the critical role in modern biology and the medicines, and we will be discussing the principle of polymerase chain reaction, and you're going to learn how the primer can be designed, and we'll understand how their applications in genetic testing or even forensics or disease diagnosis and research can be used. In the real world case studies, it will demonstrate that this technique, this technique can impact like solving complex biological problems. The content of this video will be: first we will define what is primer and its role in polymerase chain reaction, and I will discuss about the importance of primer designing, like how it can be designed successfully for polymerase chain reaction. And the important considerations for the primer designing will be discussed. We will discuss mainly about the length of a primer, why it should be of certain length. Like for example, generally it is recommended it should be between 18 to 25 base pairs, and we will discuss what are the major reasons that this particular length should be chosen, and then we'll discuss about the GC content. These are very important because they generally it is recommended at 40 to 60%. We will discuss what is this exactly mean in our successive slides, and we will also discuss how it can impact the melting temperature and the stability of the product, of the PCR product. And next we will discuss about the melting temperature, which is generally referred as TM, and we will define what is this melting temperature. We will also discuss what's the ideal range. Generally it is recommended to be 50 to 55 to 65%, 55 to 65 degree centigrade. And we will also discuss why it is important to not to have like why not to have like different why to have the similar TM value for the forward and the reverse primer. Then we will discuss about the specificity and the how we can avoid the secondary structures, and where we will be checking for unintended binding sites in the genome. And finally we will discuss about the 3' end design, where we will understand what's the importance of the 3' end for polymerase binding, and why it is recommended to have like 3' end composition, which is also referred as GC clamp. Additionally, we will discuss how to avoid common problems which are encountered during the primer designing. For example, how to avoid the repetitive sequences and the long runs of single nucleotides, and how we can avoid the secondary structure, for example, hairpin or even the formation of primer dimers. And we will see what are the tools available for designing primer, and we will discuss the primer blast in particular, which is available from the NCBI website. And we will do some hands-on experience about the primer testing and optimization, and we will examine especially we will focus on the designing a primer for specific application, for example qPCR or even RT-PCR, and we will discuss what are the primers for difficult template, for example GC rich regions or repetitive sequences. And finally we will recap what we learned or what we understood from this topic. Well, polymerase chain reaction is a revolutionary laboratory technique which has transformed molecular biology and genetic analysis since its development by Carey Mullis in 1980s. This is a powerful method for rapidly amplifying specific DNA segments, producing millions to billions of copies from single molecule. This PCR utilizes repeated cycle of heating and cooling to exponentially replicate target DNA sequences. There are three major steps in PCR. First is denaturation, which separates the DNA strands; annealing, which includes the binding of primers to target sequences; and finally, it is extension, which includes the DNA synthesis by polymerase enzyme. And the key components which are involved in this particular method are DNA template, primers, DNA polymerase, and nucleotides. This technique can generate vast quantities of specific DNA for minute samples that has made it invaluable for its application in various fields, including genomics, diagnostics, forensics, and even in for various biomedical research applications. It has a huge impact on molecular biology, and since its discovery by the Nobel Prize winner in chemistry in 1993, Dr. Mullis, Carrie Mullis. There are two different applications which has recently been explored in recent. We can see that there has been a huge surge of COVID-19, and during the COVID-19 testing, this PCR has been widely used for detection of SARS-CoV-2 viral RNA in patient samples that enable rapid and acute diagnosis of COVID-19 infections. And also in movies or in several serials, you might have observed like various forensic DNA analysis where PCR amplifies a small amount of DNA from crime scene or for evidence that allows investigators to generate genetic profiles for comparison with suspect samples or database records, and that is crucial for solving some crimes and also investigating who is the suspect. So these examples highlights the versatility of this method, that is polymerase chain reaction, and it is crucial in in addressing contemporary challenges in medicine and as well as the law enforcement. So here are the images of typical PCR machine. So on the left hand side you can see it's a PCR machine from Biorat company, and in the right side you can see there this is from the BioQuant company. So this is how a typical PCR machine looks like, and even the right side one is basically a qPCR, quantitative polymerase chain reaction machine. So in the PCR we generally amplify and we make some cDNA, and in the qPCR we can do even one step, or even we can have like amplification graph, and we can examine quantitatively what's the amount of or what's the level of gene expression has been done. So you might be wondering why it is important to design properly a primer for a successful PCR. Well, the proper primer design is crucial for successful PCR experiment because it's a well-designed primers can ensure safe specificity, efficiency, and reproducibility of the amplification process. The key considerations include optimal length. For example, generally it is recommended 80 to 25 base pair, and appropriate amount of GC content, that is around 40 to 60%, and optimum melting temperature, which is generally recommended 55 to 65%, 65 degrees centigrade. All these details we'll be discussing in our further slides. The primer should avoid complementarity within as well as between pairs to prevent primer diamond and hairpin structures. So these are the secondary structures which needs to be avoided while designing a primer. And the 3' end design is critical for polymerase enzyme to bind and extension initiation. The specificity checks against the target genome, which are essential to avoid target amplification. And the careful primer design minimizes non-specific products, and that improves yield and enhances the overall reliability of PCR results. So these are the details which need to have focus in primer design in fundamental is fundamental for amplifications in diagnostic research and forensics, where accuracy is a paramount. So here you can see a typical polymerase chain reaction plot, that is a PCR plot, which is often seen in real-time or quantitative PCR. It displays amplification of DNA over multiple cycles. The plot features cycle number on x-axis and fluorescent intensity on y-axis, and the curve typical has three major phases, that is baseline, exponential, and plateau stage. Initially, the baseline shows little change in fluorescence, and as amplification becomes detectable, the curve enters the exponential phase, where it rises sharply and doubling with each cycle. The plot then transitions into plateau phase as reagents are depleted and amplification efficiency decreases. The threshold cycle, that is which is often referred as CT value or quantitative cycle, is a key feature indicating the cycle at which fluorescence exceeds background level, crucial for quantitative analysis. Well, let us know what is a primer. So primer is a short single stranded DNA sequence which is typically 18 to 25 base pair, as I discussed before. It's 18 to 25 base pair long, and that play a crucial role in PCR. And these oligonucleotides are designed to be complementary to specific regions which flanking that target DNA sequence to be amplified. And the primers serve as a starting point for the DNA synthesis and that defines the exact region to be replicated during PCR. By binding to their complementary sequences on the template DNA, primers enable selective and repeated amplification of the target DNA sequence. They act as binding sites for DNA polymerase, initiating the replication process, and the primers are typically used in pairs, forward as well as reverse, to amplify both strands on the DNA template. The design and selection of primers are critical factors in determining the specificity and efficiency of the PCR reaction, which is directly impacting the success and accuracy of the amplification process. One of the important factor is the primer. While designing primer is its length, because that determines its specificity. Primer in this length range are long enough to be sequence specific, and that reduces the chances of binding to unintended targets in the genome. They provide a good balance between uniqueness and practicality for the PCR application. And regarding the efficiency, the primers of this length hybridize efficiently to the template DNA during annealing step. They are short enough to allow for rapid and efficient binding, which is crucial for PCR amplification. In the context of melting temperature, this length range typically results in primers with melting temperature between 55 to 65 degrees centigrade, which is ideal for most PCR protocols. It allows for consistent annealing temperatures across primer pairs, and that improves reaction consistency. In terms of synthesis and cost, this primer in this range are relatively easy and cost effective to synthesize with high accuracy. In terms of stability, they are long enough to form stable duplexes with the template at typical annealing temperature. And also it is flexible because this range provides enough flexibility to adjust for optimal GC content and avoid problematic sequences. The primers in this range tend to produce optimal amplification efficiency, often close to the theoretical maximum of doubling the product with each PCR cycle. They are short enough to minimize the risk of internal secondary structure or primer dimers that could interfere with the reaction. Another crucial factor while designing a primer is GC content. In PCR, considering the GC content of primer is crucial for several reasons related to melting temperature and stability. For example, melting temperature, in the context of melting temperature, these GC pairs form 3 hydrogen bonds while AT pairs form 2 hydrogen bonds. The higher the GC content leads to higher melting temperature, and the recommended GC content is 40 to 60%, which helps to maintain the optimal melting range, that is melting point range, that is melting temperature range, that is 55 to 65 degrees C for most PCR amplification. GC rich regions form more stable duplexes due to strong hydrogen bonding, and the balanced GC content, which is around 40 to 60%, it provides sufficient stability without being too rigid. And the proper GC content ensures efficient primer annealing to the template. Too low GC content may result in weak binding and poor amplification. The balanced GC content helps maintain specificity by avoiding non-specific binding. Also, extremely high GC content can lead to mispriming and off-target amplification. Optimal GC content contributes to consistent and efficient amplification. It further helps to maintain similar melting temperature between primer pairs, which is crucial for efficient PCR product. Also, balanced BGC content reduces the likelihood of forming a problematic secondary structure like a hairpin structure. And this 40 to 60% of GC content, it allows for flexibility in adjusting the annealing temperature and other PCR parameters. Therefore, by adhering to the recommended 40 to 60% GC content, the primer design makes a balance between stability, specificity, efficiency, which contributes to successful PCR amplification. And this range provides enough strength for primer template binding while avoiding issues associated with extreme GC contents. Let's learn some calculations regarding the GC content. So as we discussed previously, the ideal makeup of a primer is 40 to 60% GC content. Suppose if we have 20 base pair in a primer and we have 6G and 4C, So the GC content will be 6 plus 4 divided by 20 base pair, that is 50% GC content. So that's how manually even you can calculate how much GC content is there in your primer. Again, another important factor, that is melting temperature, is crucial in PCR for several important regions. For example, this melting temperature is a temperature at which 50% of DNA strands are denatured, which that means the single stranded are formed, and 50% are annual, that is double strands are formed. Therefore, the understanding this melting temperature, it helps in determining the optimal PCR condition. The ideal temperature, as we discussed previously, is 55 to 60 degrees centigrade, and this is the range which is optimal for primer specificity and efficiency. And it allows for stable primer template binding without excessive non-specific interactions. And the temperature in this range are easily achievable and maintained by most thermocyclers, as we saw previously. And the importance of this similar TM for primer pairs is the forward and reverse primers should have similar TM values, ideally within 5 degrees centigrade of each other. If the TM value differs significantly, one primer may bind preferentially and leading to inefficient or biased amplification. The annealing temperature in PCR is typically set 5 to 10 degrees centigrade below lower primer melting temperature, and this allows for efficient primer binding while maintaining a specificity. And the proper melting temperature considerations helps avoid non-specific binding and primer dimers. It ensures optimal primer template interactions that leads to efficient amplification. It has been found that the understanding of this melting temperature, it allows for fine tuning of PCR condition to improve yield and specificity, and the melting temperature considerations guide primer design, helping to select optimal sequences for PCR. It further helps in troubleshooting the PCR issues related to primer binding. Therefore, careful designing or considering this melting temperature in primer design and PCR setup, it is very much improve the success rate and efficiency and specificity of their PCR reactions. And this attention to melting temperature, it helps to ensure reliable and reproducible results in molecular biology experiments. Again, although there are many different tools or online softwares which are available to calculate this melting temperature, but for manual calculation, it is quite useful to understand there is a trick that for that 4 degree C for 4 degree centigrade for every G or C, and 2 degree centigrade for every A or T. If you consider that, for example, here you can see that this is the sequence, this is the primer design, and where we have like so many GCA, GTA, GCA, like this. And for every G, if you consider 4 degree C, and for every A or T pair, if you consider 2 degree C, then the overall your primer melting temperature will be 58 degrees centigrade. So that's how even you can calculate like depending on the primer length. If you have a particular primer length and how many G and C, if you know, suppose if you are given with the GC content, and if you are asked to calculate what will be the melting temperature, you can reverse calculate using this technique manually. Well, another important parameter which needs to be considered is to avoid the secondary structure formation, which gives the specificity in the PCR primer design, and it is crucial for several important reasons. For example, to avoid the complementarity within and between the primers, and that prevents primer dimer, because the complementarity within a primer or between primers can lead to primer dimer, where primer binds to each other instead of the target sequence, and this reduces PCR efficiency and can produce unwanted products. It improves the amplification efficiency. When primers don't bind to themselves to each other, the primers are available to bind to the target sequence, and that increases the amplification efficiency. Again, reducing the background noise is quite important, because minimizing the primer dimer helps reduce the background amplification. If there are some unspecific or non-specific target which can be bind to this your primer, then it will create a noise, and that will impact your PCR product. And this therefore it improves the signal to noise ratio in the PCR results. Again, checking for unintended binding sites in the genome is important, because by checking for potential binding site elsewhere in the genome, it can avoid primers that might amplify unintended regions, leading to non-specific products. Also, it improves the accuracy, because specific primers that only bind to the intended target sequence can produce more accurate results, specifically important in diagnostic and quantitative PCR amplification. Further, it reduces the false positive result, because the primers that bind to multiple sites in genome can lead to false positive results, and particularly it is problematic in diagnostic tests or expression studies. And it enhances the experimental reliability, because the specific primer lead to more consistent and reproducible PCR results across different experiments and laboratories. Another important aspect is the 3' end design of a PCR primer, which is crucial for several important factors. For example, the importance of this 3' end for polymerase binding. It acts as an initiation point. The 3' end is where DNA polymerase enzyme initiates the DNA synthesis, and proper binding at this end is critical for efficient amplification. And a stable 3' end ensures the polymerase can attach and begin extension without dissociation. The 3' end largely determines the specificity of primer binding. The mismatches at the 3' end can significantly reduce or prevent amplification. The recommendations for 3' end composition, which is often referred as GC clamp, is including 1 to 2G or C bases at the end, which is known as GC clamp, and it can improve the primer binding stability. This prevents the mispriming or formation of primer dimers. AT at the ultimate 3' base can lead to mismatches more easily than others. There are additional considerations which can be followed. For example, 2 to 3 bases at the 3' end should not be complementary between primer pairs to prevent primer dimer, and we should also avoid mismatches between 3' end of the primer and template, as this can significantly reduce the amplification efficiency. Further, we will do some practical exercise where we'll do the hands-on primer design for some sample gene sequence, and we will evaluate the design primers using software tools. So these are the common steps which we follow while designing a primer. First is we will select our target sequence. We'll identify the sequence DNA region which we want to amplify. We will obtain its nucleotide sequence from the database like NCBI. We'll choose the primer location. We'll select 18 to 30 nucleotide at the 5' to 3' ends of our target region. And for reverse primer, we use the reverse complementary sequence. And we will analyze the primer properties. For example, as we discussed previously, we will check for the GC content. I will calculate the melting temperature, and we will ensure the forward and reverse primers have similar melting temperatures or within the 5° centigrade variability. We'll try to avoid the problematic sequence, as we discussed before. Like, we will try to avoid the form so that it will not form the secondary structure. We'll check its specificity by ensuring the primer sequence is unique to target region. We'll use tools like BLAST to check for unintended binding sites. Finally, we will do some primer validation using some in silico softwares, and we will design our own experiment. So in general, these are the tips for primer design. Although we studied in much detail before, but these are the common take home message. Like, primers should be as pure as possible, around 17 to 30 base pair length, and the base composition should be 20 to 70% GC content. Whenever possible, avoid the unbalanced distribution of GC and AT rich domain, and the primer should always end 3' in G or C or C or G or G or C. This prevents breathing of ends and increase efficiency of priming, or try to make GC content equal in both primers. The melting temperature between 55 to 80 degrees C. So and the 3' end of the primer should not be complementary, as otherwise the primer dimer will be synthesized preferentially to any other product, and that can cause non-specific amplification of the target, non-specific target. The primer self-complementary. It has the ability to form secondary structure such as hairpin if they can merge with each other, or even they merge with the similar the complementary primer. That should be avoided. So Runs of 3 or more C's and G's at the 3 ends of the primer may promote mispairing at G and C rich sequences, so this should also be avoided. We should optimize the concentration of formamide, DMSO or twin or ammonium sulfate. So these are the chemicals which we generally use in while doing our PCR our experiments, so we should optimize their concentration if we don't get the product. So we will be using this NCBI tool, which is a primer blast tool, which is given by this NCBI website. So we will be using this particular website for designing our primer. So this is how the NCBI website for primer blast looks like. This is the interface. I have prepared a separate video for you. You can watch that in detail. So here you can fill all the sequence of your target gene, and then there are many parameters which we need to select. So that I will be discussing in a separate video. So this is how you will get the result section why after designing the primer through the primer blast website at NCBI website. And this is the typical interface where you will get all those design primer and you will know the details. So you can watch the another video to understand how you have to do the design primer design.